Hello everyone, welcome back. Today I'm here with this 13th part of this questionnaire, and in this part I will discuss some questions from plant physiology. Let's get started. So the first question is, which of the following lacks phytol chain? So yesterday I've discussed about the chlorophyll structure of chlorophyll. So it consists of a tadpole larva-like shape, which is formed of head and tail. So this tail is made up of phytol. So our options are chlorophyll A, chlorophyll B, chlorophyll C, or pheophytin. So its right answer is chlorophyll C. This chlorophyll C lacks this phytol chain. So this is the structure of chlorophyll A, and it consists of two parts, that is phytol tail and this porphyrin head. This is made up of 4 pyrrole rings along with center magnesium. So these are joined with magnesium through this bond, which are known as coordination bond. The next, this is pheophytin. Pheophytin also contains this chain that is called phytol chain, and this is the structure of chlorophyll C. So this chlorophyll C lacks this phytol chain. The next question is substrate for ribulose 1,5-bisphosphate carboxylase oxygenases. This is Rubisco, which is the most abundant plant protein on this Earth's surface. So its substrate is 3-PGA, glycolate, RUBP, or oxaloacetate. So its right answer is RUBP, that is ribulose 1,5-bisphosphate. It works on this along with this carbon dioxide or oxygen and give rise and forms the PGA. That is the first stable product of C3 cycle, and it is made up of 3 carbon atom. So that's why this is known as C3 cycle. So its substrate is ribulose 1,5-bisphosphate. As we can see here, this is Rubisco. It works on this substrate, that is ribulose 1,5-bisphosphate, and in the presence of carbon dioxide, it combines with this 5 carbon atom sugar and forms the 6 carbon intermediate, which is unstable and is broken down into 2 molecules of this 3 carbon compound that is called 3-PGA, that is 3-phosphoglycerate. So this is the first stable product of this C3 cycle, hence it's named C3 cycle, and discovered by Kelvin, so hence its name is Kelvin cycle. It consists of 3 steps. First step is carboxylation, that is addition of carbon dioxide to the substrate, that is RUBP. And then second step is reduction. So here it involves reduction of this PGA into, that is PGAL, that is phosphoglyceraldehyde, acid to aldehyde. Then third step is regeneration. Means regeneration of this RUBP. So this is the major process of formation of a food material, that is glucose or finally starch in all the plants. Next we have the C4 cycle. So in C4 plants, they have some anatomical differences in their mesophyll cell. So they'll have differences in their mesophyll cell and bundle sheath cell. This mesophyll cell lacks Rubisco, while this bundle sheath cell contains Rubisco. Here this mesophyll cell, it contains one enzyme that is called PEP-case, that is phosphoenol pyruvate carboxylase. So its only function is to addition of carbon dioxide to this 3 carbon compound, that is it combines with this 3 carbon compound, that is PEP. So here PEP combined with CO2 to form this 4 carbon compound, that is oxaloacetic acid, which is the first stable compound of this cycle, that is OAA. And this process is done in the mesophyll cell. Then finally there is some differences in mesophyll and bundle sheath cell. So after that it is converted into another 4 carbon acid, that is called malate, and then it is transferred to the bundle sheath cell, where it undergoes the process of decarboxylation. And malate undergoes the process of decarboxylation, means यहाँ पे carbon dioxide release होता है and again converted into 3 carbon compound, that is pyruvate. It will come back to this mesophyll cell, while the CO2 is utilized similar to that as in case of C3 cycle. It will undergo Kelvin cycle and produces the starch. So this is how the C4 plants help do the carbon dioxide fixation. So the another difference is this bundle sheath of C4 plants resemble one thing that is called rith. and it is also known as Crane's anatomy. So Crane's anatomy is shown by C4 plants. The next we have this CAM metabolism. So I will discuss about this CAM metabolism. It is shown by the member of family Gracilaceae and was observed firstly in Graciola. So here these are grown in the warm humid conditions. These plants grow in warm humid conditions like xeric environment. So they open their stomata during night time. So stomata are open during night time, और जो atmospheric carbon dioxide है वो fix होती है night में when stomata becomes open. And here it shows the similarity with C4 cycle. This carbon dioxide mixed with water to form this bicarbonate ion. Then it combines with this 3 carbon compound, that is phosphoryl pyruvate, and is converted into 4 carbon acid, that is called oxaloacetic acid. Then further it undergo dehydrogenation process. It is converted into another acid, that is called malic acid. It is stored inside the vacuole. During night time this process is done, and when there is light or during daytime, stomata becomes closed. Stomata gets closed, and there is no entry of carbon dioxide or exit of carbon dioxide out of the cell. So here this malic acid comes out of this vacuole during daytime and undergoes a process of decarboxylation, that is removal of carbon dioxide. It releases one molecule of carbon dioxide and pyruvate. Then pyruvate is again converted into starch, and this CO2 is. Next question is, photolysis of water in photosynthesis require the presence of. So it is basically a question asked about this OEC, that is oxygen volume complex. So it consists of manganese ion, magnesium ion, manganese and chloride ion, and potassium and chloride ion. So its right answer is manganese and chloride ion. So these two ions helps in formation of this OEC, which is associated with photosystem 2. ये एसोसिएटेड होता है फोटोसिस्टम 2 के साथ, और ये हेल्प करता है वाटर की स्प्लिटिंग में to produce oxygen. So photolysis of water requires this manganese and chloride ion, and it releases this oxygen. So hence 4 molecules of water is required for evolution of 1 oxygen molecule. So these two ions plays very important role in the formation of this OEC, that is oxygen volume complex, and splitting of water. Next question is, value of RQ is more than 1 when respiratory substrates are. Our options are carbohydrate, organic acid, fats, proteins. So its right answer is organic acids. So the value of RQ is more than 1 or unity for organic acids. Firstly, what is RQ? So RQ is a measure of ratio of carbon dioxide produced and oxygen consumed by an organism in a per unit time. So RQ is volume of carbon dioxide evolved or produced and volume of oxygen consumed per unit time. So that is called RQ, respiratory quotient. If we talk about its value for simple carbohydrate sugar, like glucose, so here it requires 6 molecules of oxygen for its consumption. So it produces 6 CO2. So 6 CO2 are evolved and 6 CO2 are used. So its value is unity, that is 1. So for carbohydrate, its value is 1. If we talk about other respiratory substrate, like we have this fatty acid. So fatty acid contains lesser oxygen. So it requires more oxygen atoms to break fat down into the usable substrate. So its value is less than unity, that is 0.7. Again, its value is less than unity for proteins also, but its value is greater than 1 for organic acids, because acids contain more number of oxygen. So that's why its value is more than 1 for organic acids. On the basis of respiratory substrate, we have different types of respiration. First is the floating respiration, where the respiratory fuel is carbohydrate and fats, while it is known as protoplasmic respiration when the fuel is protein. So these terms are also used for respiration on the basis of the respiratory substrate used. The next question is, which of the following is not a component of mitochondrial electron transport chain? Our options are ubiquinone, cytochrome B6, cytochrome C, or cytochrome A and A3. So its right answer is cytochrome B6. This cytochrome is involved in light reaction. It is not involved in mitochondrial electron transport chain. So cytochrome B6 F complex is an enzyme bound thylakoid membrane. It is found in chloroplast, a green plant that catalyzes transfer of electron from plastoquinone to plastocyanin. While other options, like we have ubiquinone, cytochrome C, cytochrome A and A3, these are part of electron transport chain, that is mitochondrial electron transport chain, and these are present in inner mitochondrial membrane. Cytochrome C helps in transferring electron from complex 3 to complex 4. This is the role of cytochrome C. And cytochrome A and A3 transfer electron and catalyze the reaction of oxygen to water. And ubiquitin is a mobile component of the mitochondrial electron transport chain. It acts as a membrane antioxidant and also receive electron from both the pathways, that is one from the NADH, another from the FADH. So this is the electron transport chain, mitochondrial electron transport chain. And here we can see here this ubiquitin is involved. Then we have cytochrome C and cytochrome this A and A3, but cytochrome B6 is lacking here, so this is not present here in the mitochondrial. So this was all about for our today's discussion. Hope you get some idea from this presentation. Have a great time.